beginning
folder,
of
the
5th
general
lesson,
folder,
Gr12,
because
please.
I stopped
1 and 2,
uploading
30th of
videos
July,
last
start.
week. Sorry, I stopped uploading videos last week, so Mam's already uploaded lesson 1 and 2, but we're still waiting for, I think yesterday's, is it the day before as well? No. Yes, yeah, it's yesterday's, and we'll wait for today's link. We need to wait for it to come from Mr.
I put yesterday's up yesterday.
Oh, is that already that one? Oh, I have no idea what date it is. Okay. Oh, because we had Monday and Wednesday. No, you're right. We didn't have Tuesday. You're right. So, okay, so today's is going to go up here, and then I just added some general evolution videos. Now we'll watch 1 or 2 of those just now, and then at the top here, those are all the other lessons from last week, and Mam's other lessons from last week, and what to study for prelims, revision. Did you all look at that when you revised for the test? So, yeah, see some of you doing this. Okay, and you've got to go to Moodle, I said, and that's the question paper, info, okay, going over model. Alright, so I've just uploaded the Word document here now, just to make it easier for me to zip through it. Test, hang on. Yeah, this one. And oh wait, is it open or is it blue? Oh, I want a split screen. I don't want to open use that. I just like it when it does this. Where's download? Okay, it's not doing it. So then I'll just use it from the top here. Okay, so we got up until, I'm going to have to flip between screens. Alright, we got up until, yes, we got up to here. Alright, so please, those of you who weren't here, you've got to go and look at this question. And I was saying that you can't just read. You must cross and tick and see which one's right. You can't just read sort of and not make any marks on this question paper. Yes?
So this morning when you were speaking about the inner implication, is there any further developments if we said A, it occurs before Mitosis 1 and 2 because of the wording of that is a bit funny?
Yeah, but this, I think I got this from a past paper. So they're saying here, well, I was also saying it occurs before Mitosis 1 and Mitosis 2. No, it only occurs at the beginning of Mitosis.
No, I'm aware that
okay.
in order for Mitosis 1 and 2 to occur, that needs to happen before it. So because of the wording, it looks like it's one entity, and then before that you have...
Ma'am, what do you think? I'm seeing it my way, as it occurs at the beginning only. Okay, yes.
I think it's just because of like the wording, and because there were so many people who understood it that way, that means that there is a very high likelihood that that's the way that it came across to most people, and then the wording is therefore ambiguous.
What if you get it like this in November? Then what are you going to do?
We'll fight again.
No, you won't. You won't, because you won't know. You won't know if you got it right or wrong.
I'm just saying.
Okay. All right. So bear this in mind. The way I read it, the way I've seen it before, is that it means before both of them know. Because meiosis is one entity. Okay? It's one process. So DNA replication occurs before the one process. But if it's written like that, it's implying that it occurs before meiosis 1 and before meiosis 2. So I'm going to leave it there, because then I'd rather you remember that than go and, they give a similar question or whatever at the end of the year, and then you're still thinking, you know what I mean? You know what I'm trying to say? I'd rather leave it like this. Because that's the way I see it. It occurs not before both of them, only at the very beginning. And to me, that's at the beginning of both of them. Okay. All right. So rather let's leave it there, and then you're very aware of it. All right. So, okay. Meiosis. How happy are we with meiosis? When we think about all the stages, I can't remember the individual what we got here. Who is superb with meiosis? Who's average? Who feels confident with it, is what I'm saying. Who feels, not really, okay, so you're right. So just, okay. So yes, it occurs in specialized cells in the ovaries or the testes, okay? The cells divide by meiosis to produce gametes, and yeah, it's 4 again. I didn't really want another diagram where the diploid number was 4 again, but it's okay. All right, so this cell, the cell that was diploid had been, the cell which underwent meiosis had a diploid number of 4, and then these are different stages. Now, most of you got the stages correct, but I saw that a lot of you switched two of them around. Okay, so if we were looking at this, before we even look at the questions, how do we know which ones are meiosis 1? How do we know that whatever's happening is meiosis 1? There is one cell. What else? The chromosomes are still? They're still like that. So put that in words. They're still replicated. Because the purpose of meiosis 1, sometimes it's called reduction division, yes? So the purpose of meiosis 1 is to reduce what? Reduction division, reduce what? Come on, guys. The initial cell was diploid. So the purpose of meiosis 1, if the initial cell is diploid and there are 4 chromosomes. Okay, let's go back. Mitosis. If your skin cells are replacing skin cells, because you're always replacing skin cells, they've always been lost. Mitosis is to do what? What is the function of mitosis? Simply, mitosis is it allows for. Come on, guys, you know it. Loud. I can't hear. Growth. Okay, so at the end you're going to end up with more diploid cells. Yes? Okay? Meiosis, we're trying to produce gametes. We're trying to produce more gametes. Alright, so meiosis 1, the first division, I think I've got a pen on my jacket, it's known as reduction division. Why? What does that mean? It reduces what? Number of? Yes, come on, I can't hear you. Chromosomes. Yes, it reduces the number of chromosomes. So here you have one cell. By the end of meiosis 1, how many cells are there? Sorry? 2. Come on, guys. There are 2 cells. Here they were diploid. They had 4 chromosomes. Here they will be haploid. So you've got 2 times haploid cells. Alright? Then each cell undergoes meiosis 2. Okay, so meiosis 1 is called reduction division. It reduces the ploidy. Alright? Meiosis 2, so each of these daughter cells now undergo meiosis 2, which is similar to what? You know this? Similar to mitosis, because you're just increasing cell number, but the ploidy stays the same. So you end up with 4 cells, and what ploidy will they have? Sorry? Haploid. Okay, so 4 times haploid cells. Okay, it's looking a bit squashed here. Alright, so okay, let's try and find another color here. Alright, so if the original diploid cell has got 4 chromosomes, okay, when the cell's in interphase, the DNA is in the form of what? When the cell's in interphase, the DNA is thread-like, we call it chromatin. Then it coils up, we call it chromosomes. Then what happens? Replication. So you end up with, okay, let's go back. Alright, how many DNA molecules here? Forming that strand of chromatin. How many DNA molecules? I'm just looking at this. 1 DNA molecule. When it coils up, how many DNA molecules? Still 1. When replication occurs, now how many? 2. Okay, but hang on. So here we've got 1 strand of chromatin. Here we've got 1 chromosome. How many chromosomes there? 1, because you count centromeres. But it is what type of chromosome? It's replicated. Alright, so over here, Okay, so 2n equals 4. So at the beginning, in interphase, how many strands of chromatin am I going to draw? The diploidy of this cell is whatever. I don't know, Oompa little organism. It's going to divide, cells are going to divide by meiosis. Its ploidy is 4. How many strands of chromatin am I going to draw? 4. Alright, now it's getting ready for meiosis. What happens to those strands of chromatin? Okay, coil up and replicate. Alright, so it's going to end up with 4 coiled up strands of DNA. Okay, so do we understand that whether it's chromatin or chromosome, it's DNA, it's made of DNA? Yes? So what's this table made of? Wood. What's that lab still on the side made of? Wood. But they're different. One is a table, one is a chair. Got it? So here, chromatin and chromosomes are both made of DNA, just in different forms, yes? Alright, so now the chromosomes have coiled up, then what occurs? Sorry? Replication. Alright, I think I'm drawing very small now. Okay? So, okay, go back. How many strands of DNA at the top? How many here? How many here? How many strands of DNA? 8. Okay. There's 4 chromatin strands there. How many chromosomes? How many chromosomes? Replicated chromosomes, because you count the centromeres. I think I'm going somewhere with this. Alright, so when the cell divides, how do these chromosomes line up along the equator? With their in homologous pairs. So maybe I should have drawn 2 of them longer and 2 of them short, just like the diagram. So they're going to line up in their homologous pairs. Alright, in that cell. Okay, then meiosis. Whoa, sorry. Then meiosis 1 occurs. So how many am I going to draw in each cell? Reduction division. How many will I now draw? If this diploid cell undergoes anaphase, telophase, etc., I end up with 2 cells. How many chromosomes each? 2. How am I going to draw them? I've got 2 long, 2 short.
1 big, 1 short.
1 big, 1 short.
1 big, 1 short.
1 big, 1 short. What's the ploidy? N equals? How many chromosomes per cell? Do you agree? It's 2 chromosomes. You count centromeres. They're just still replicated. They haven't said goodbye. Okay, the chromatins haven't said goodbye yet. So N equals 2 chromosomes. Okay, so now each of those divide by meiosis 2, which is similar to mitosis, producing more cells. Alright, so how will the chromosomes line up now in each of these cells? In a single line. Long one, short one. Now imagine if we've got 23 chromosomes. They're all going to be lined up along the equator. But don't always revert back to 46 and 23. Because I heard someone saying 46 just now. What is the question asking? Are they asking you to look here? Or are they asking you about human cells in general? We all want to talk about human cells. Okay? Alright, so there they're lining up singly. So now, if both of them are doing that, okay, so both of these cells, the chromosomes are lining up along the equator, single file, and I'll get to the question now why I didn't give marks, okay? So now what happens? Now it's meiosis 1. So what happens to each of those chromosomes? What's holding the 2 chromatids together? The centromere. Maybe we should draw it better, okay? So now, there's the 2 chromatids, sister chromatids. The centromere splits, and what moves into daughter cells? Okay, let's just take one of them down. So that becomes eventually. Alright, let's draw anaphase first. Okay, so now, something like that. Okay, so that's those centromeres have split, and now what gets pulled to opposite poles? What were these 2 called? But now we can call them or daughter chromosomes, because they are going to separate into daughter cells. So you'll end up with 2 daughter cells from that cell with how many chromosomes each? Look. 2, this one's got 2, and the same for this one over here. You're going to end up with 2, which is, I thought it was drawn there. Okay, are we following? Right, so go back to the beginning. 2n equals 4. There are 4 replicated chromosomes. Meiosis 1, the pairs split, homologous chromosomes separate. So you end up with 2 daughter cells, but they're haploid. They've got 2 chromosomes each. Yes, they're still replicated, but a replicated chromosome is still considered 1. It's just got a copy. It's got copies from DNA replication. Then each of those daughter cells undergo meiosis 2. So the result is, if I had to now draw the chromosomes in each of these daughter cells, what am I going to draw? So there I had a long and a short, and a long and a short. What am I going to draw here now? Got on the boards there. A long and short single one, like that. Okay. So now let's match it to these pictures over here. Alright, so it says, okay, using the letters P to S, put the diagrams in the correct order to show the sequence in which the stages of meiosis would take place. So my first question was, if it's meiosis 1, how many cells would you see for most of the process? If it's meiosis 1, you're going to see 1 cell. What else are you going to look for? Chromosomes which are paired. Okay, they pairs and then the pairs separate and so on and so on. So, is P meiosis 1 or meiosis 2? Pairs are separating. Is it meiosis 1 or 2? 1. Okay, is it what stage of meiosis is it though? Anaphase? 1. What is this? Q. How many cells do you have? 2. Alright, so remember, 2 cells undergo meiosis 2. Alright, so that is meiosis 2. What stage of meiosis 2? Because you're not separating paired chromosomes anymore. They now, I've got that out. They've lined up singly along the equator. Alright, stage R. Meiosis 1 or 2? 2. Because those 2 daughter cells, their chromosomes are lining up singly, or they did line up singly along the equator, and their chromatids, or daughter chromosomes, are now being pulled to opposite poles. So which stage is this? Anaphase 2. What is S? Because they've coiled up and replicated, it could be coiling up, replicated, prophase. They just haven't drawn nuclear membranes and all of that. So expect diagrams where it doesn't always look like you had it. So that is prophase 1. So if you just wrote prophase or anaphase or metaphase, I can't give you a mark because you need the 1 or the 2. Or else you're talking about mitosis. Okay, so put them into order. Which one is first? S, prophase 1. Then? Sorry? Yes, because the pairs are being separated. What stage is that? Anaphase 1. Then? Q, because they're lining up singly along the equator in each cell. And that stage is? 2. And here? Anaphase 2. So it's S, P, Q, R. Okay, yes.
Can R not be telophase because it's starting to... No.
Yeah, they've shown... oh, no, hang on, because it's 2 cells. Yeah, if it was telophase, cytokinesis, then you get the indentation. But remember, telophase is when those chromosomes have moved to the poles and nuclei have formed around each of them, and then cytokinesis is when the actual cell divides. Okay, so telophase, I say 2 nuclei form. Alright. Okay, so let's have a look now. Identify the meiotic stages shown. Okay, I think we've done that, but P, remember that was? What was that again? Anaphase 1. But now you've got to give a reason. So can you just say, because the chromosomes are being pulled to opposite poles? No. What's different here? It's not mitosis, where chromosomes are pulled to opposite poles. What's being separated here? What are these? Homologous pairs are being pulled to opposite poles. If we look at anaphase 2, how is it different? You can't just say because chromosomes are being pulled to opposite poles. Here, sorry? Centromeres, yeah, centromeres, yes, because here there is no centromere splitting because the whole chromosome, the whole replicated chromosome moves to the poles. Okay, there's no separation or no division of centromeres. Whole chromosomes get pulled to the poles, but in anaphase 2, the centromere splits, and what moves to the poles? Chromatids, or I call them daughter chromosomes because they're going to become part of the daughter cells. Okay, so you've got to give something that is visible. Alright, so P, it's anaphase 1 because homologous pairs are separating and moving to the poles. And then I asked for Q, and Q we said is? So you can't just say chromosomes lining up along the equator. How are they lining up? They're not lining up in pairs, they're lining up? Sorry? Singly. They're not lining up in their homologous pairs, like I've drawn there. They're lining up singly along the equator, because the pairs have already been separated. They're lining up by themselves singly along the equator. Alright. Let me stop there. Okay, let's just go to the model quick.
End of for 5th grade,
I don't know where I am.
12, choice 1 and 2, 30th of July.